The sooner the better. We get early here, out of here. So last time I told you about long-term potentiation and long-term depression as a great mechanisms for learning and memory. And this class, I'm going to tell you that long-term potentiation and depression are not the whole story. That you need two more processes, or to understand it in two different ways. And one, so what I'm going to talk today is about spike time independent plasticity, and this is a better model, actually, of how plasticity may really happen in the real behaving brain, and also the notion of homeostatic regulation of synaptic plasticity, that LTP and LTD, even if they're so good, you cannot let them run free. You have to control them, otherwise horrible things happen to you. Okay, so I'm gonna start by recapitulating a little bit what we saw last time. And so now we know that long-term potentiation and long-term depression is basically initiated by the activation of the NMDA receptors, you know, by the glutamate, postsynaptic depolarization. If you have a big jump of calcium, you activate kinases, and somehow that is good for LTP. And if the sip of calcium is kind of moderate, you activate phosphatase, and somehow that's good for LTD. And there's a pretty standard consensus now about that. And okay, and the how you can say the part of the evidence of that is that you can vary the parameters of the stimulation and get a smooth curve going from LTD to LTP. So for instance here, if we vary the frequency of a stimulation, you give like a lot of pulses, like hundreds of pulses, but you give it first at low frequency, you get nothing, very low frequency. But if you give it like no calcium basically here, at 1 hertz, as I told you, for 15 minutes, you can get LTD. But you give now the same number of pulses, but a higher frequency, you get more and more LTP. So you have this kind of like a biphasic curve. Okay, a region for LTD, the moderate activation, and higher activation is going to be LTP. And that's probably reflect the amount of calcium. And you can do the same thing by giving now the same number of pulses at the same frequency, but you can control the postsynaptic, the voltage of the postsynaptic membrane. Okay, so if you don't do anything, you don't get nothing. But if you depolarize a little bit, from normally they're sitting at -70 or 80, and you depolarize to -40, then you can get LTD. But now you depolarize more, you get a crossover point, and eventually if you depolarize a lot, like a -20, you activate now the NMDA receptors a lot, and then you can get LTP. So you have, again, this is a very common motif, like the biphasic two regions curve. But that's very good for inducing LTP and LTD in the lab. But probably that doesn't happen in real life. So 100 hertz for a long time, that's epilepsy. Okay, so you don't need epilepsy to learn things. You learn things without epilepsy. 1 hertz for 15 minutes. That's the most effective way of inducing LTD in the lab. Is not probably not a realistic, okay? Unless you go to a disco and you get like a bump, but probably you're gonna get a little bit depressed. But and on the other hand, this prolonged depolarization for like several seconds. So if you depolarize the cell to 0 millivolt or to -20 for like 20 seconds or a minute, that's actually coma. Okay, so that you get this situation normally when you're in a coma, not when you're learning. When you're learning, you get just action potentials everywhere. So we need a model of inducing plasticity more or less based on the firing of the cells. Okay, and people have thought about that, and they're thinking like, you know, if you have two sets of input to this cell, this will be a pyramidal cell in the cortex, and more or less they're firing at the same time, each one of these will be an action potential. And actually, this is a representation of a real, actual experiment. So this is the visual cortex, and these are the input that eventually coming from the two eyes. Okay. So when the two eyes are, you have two eyes. They're always looking at the same thing, more or less, a little bit. So you're having kind of correlated activity between the two eyes. the inputs from the two eyes, and make the postsynaptic cell to fire, and everybody's kind of correlated. Okay, so everybody's firing together, and that should be good for inducing potentiation. Okay, so this is a good correlation. On the other hand, this is an experiment that people have done. If you close one eye in a very young mammal for like several days, although you have to be like, and then you reopen that eye get disconnected from the cortex. Okay, it's a classical experiment of plasticity when you are very young. Okay, that terrible thing happens. So you don't close your eyes too long when you're young. You're older now. But anyway, so what happened is that the closed eye is producing kind of like a spontaneous activity, which is totally uncorrelated with the postsynaptic cell in the cortex. And that is somehow driving the disconnection, the depression. So basically, non-correlation and anti-correlation, kind of like a Gunther Stent postulate. Remember the Hebbian postulate that this cell A fire and makes cell B fire, that should be potentiated. Well, that's the case on here. And the stent, which is the anti-thing, is like, if cell A in this case is not making the cell B to fire, that should be disconnected. Okay? And it turned out that that happened in real life. Okay? And the way that people discovered that, it was kind of like a little bit of a tortuous way. So it came, first of all, it was initiated basically by this early experiment in the 90s, in which these two guys, Sackman actually get a Nobel Prize for another thing, for discovering some recording and channels, and then he decided to go to the cortex and study this thing. What they did is they put an electrode in the soma, okay, and they polarize the cell to produce an action potential, which is going to be this blue thing in here. Classical action potential. And then they record, here's the axon. So the action potential normally is initiated here as the so-called axon hillock, and it propagated here. And then, of course, in the axon is also the same action potential, perhaps a little bit bigger. People know that for like hundreds of years. And everybody thought that the action potential is initiating here and go this way. What this guy found is actually no, it goes both ways. It can also go into the dendrite, and it's called backpropagation. People have never been able to see that, because what you had to do is you go with an electrode and record from the dendrite. That was very difficult, but these guys managed to do it, okay? And this was a seminal paper like 30 years ago, and they showed that in the dendrite, you can get the same action potential, a little bit kind of sluggish and kind of like a more wobbly, but still it can go all the way very up, okay? So you have the backpropagation of the action potential. So all of the synapses in here, they get informed that something very good happening here, basically. That this guy produced an action potential. And actually, it turned out that backpropagating action potential is crucial for inducing LTP. Okay? So here, later, they are doing this, an experiment in which the cell soma is in here, or these other people, okay, and they're recording and they're inducing action potentials, okay, these bursts of action potentials, okay, and they go and they put actually a fluorescent dye so they can see where the dendrite goes, and they go with another very small electrode in here to stimulate synapses only in this region, okay. So what they do is do the synaptic stimulation, action potential, they're kind of pairing those, okay, in close in time. And when they do that, and this is the responses get bigger. If they do unpair, so like say a whole bunch of action potentials first, then wait a couple of minutes and then give all of the synaptic stimulation, nothing happened. But if they do it like at the same time, more or less interleave, like in here, they get potentiation. Okay, so and it's the same kind of stimulation. So the idea is that these action potentials are traveling all the way there, and helping, fulfilling the Hebbian rule. And this is a refinement of that experiment. What they're doing here, the same thing. They're recording here, illuminated with fluorescent, the whole thing, and here they are recording. They're stimulating in here, and they're doing two things. They're measuring actually calcium signals. So they use the calcium signals to see, how much whenever you have an action potential, you open calcium channels and you leave a calcium inside. So by recording the calcium signal here, they can see how well the action potential is propagating. Okay? And they're going to do something very cool, which is going to put TTX in here. TTX blocks sodium channels, so they should block the propagation of the action potential. Okay, so this is and what happened is actually, I forget to tell you, the dendrites do have sodium channels just like the axon. Okay, but not in that amount. Okay, they have a lesser sodium channel, but sufficient to allow some propagation of the action potential. Okay, so they're going to block those sodium channels. And what's happened now? So when you do that, this is one. Where is one? One in there. Okay, so this is the normal signal that you get when you're firing the signals on the action potential. But if they put TTX in here, all right, they don't get any increase on calcium when you fire the action potential, because the idea is that the action potential don't go farther. So you get they don't propagate it to 1 or to 2, but you still have the calcium signal in 3 and 4 because it's below that. And more importantly, when you put the TTX and then you repeat, you give the action potentials and the synaptic activation in conjunction, you don't get anything. Okay? So this is the response when you pair action potentials initiating here plus synaptic activation in here, but if you put the TTX, nothing happened. And the idea is that because the action potential couldn't go all the way up. All right? But now you wait a little bit, and then the TTX is gone, and now you repeat it, and then you get the LTP. So if the action potentials are able to go all the way up to there, then you have it. Okay? And they work a little bit on the other people, of course. This is just people after people. And this is a two-photon imaging, one of the early two-photon imaging, but they demonstrate something very interesting. So they're imaging the calcium inside the spine, one of those, okay, with the fluorescent techniques, okay, so you can do that. And if you stimulate locally here, you get a little bit of a calcium increase. Probably some activation of NMDA receptors, okay? Moderate. Then you produce action potentials, okay, and action potentials, and you can also detect the increase in here. But now you can activate, you produce the action potential and activate the signal simultaneously, or close to simultaneously, and you get a much bigger response. But importantly, it's supralinear, because if you add this plus this, you get this, okay? So it's a superlinear interaction. They help each other, so the action potential is really actually helping the NMDA. And it kind of makes sense, because the NMDA receptor, in order to be fully active, need to be depolarized, okay? So the action potential goes all the way here, and this guy already get the glutamate, but if the action potentials arrive there at the right moment, then it helps to remove the magnesium and fully activate the NMDA receptor, okay? And on working on that, there was some interesting twist of this, that the order of whether you produce the action potential and the synaptic stimulation is very crucial. So if you activate the synapses, give it tum, 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 and then you give the burst of action potentials, or one, in that case you get this big response. Now if you give the same number of synaptic activations and the same number of action potentials, but in the other order, you don't get it. Okay? And the idea is very simple. So if you activate the synapse, the NMDA receptors are going to get the glutamate, and the glutamate is going to be there for a while, for about like 30 or 50 milliseconds. So if at that moment you came with the action potential and you depolarize, and then you relieve the magnesium block, then you can fully activate the NMDA receptor. Okay? The other way around, it doesn't work, because the action potential is very fast. So if you give the action potential, the whole thing is going to be depolarized for like about 10 milliseconds, and then...
Do these interactions happen normally?
Huh?
Do these interactions happen normally, or is it just a rare occurrence?
Well, this is an interaction that happened in a slice of a mouse brain, you know. you cannot observe this in the real thing. But the idea you will see it is that we think that that's the way it happened. Okay, there's many indirect evidence, like a wealth of indirect evidence that probably that's what happened. But you cannot observe that directly in vivo. Okay, but the idea is that you have an asymmetry, a temporal asymmetry. In one way, you get the big activation of the NMDA receptor, and in the other one, you don't. And that is getting close, actually, the fulfillment of the Hebbian and the anti-Hebbian rule, because in the Hebbian rule, they say this is cell A fire, okay, and then cell B fire, you get a potentiation, and the other one, if you cell B fire first, and then you get the synaptic activation, you should get depression, okay? And this thing with the backpropagation and the activation of NMDA receptor is kind of like a mirroring this thing, okay? So it's kind of tell you that perhaps the only thing that you have to care is the order of who get activated, okay? And that's what people can observe, and actually there is evidence. I don't think I'm going to have time, but that can happen in humans. Okay, but here it is, an experiment in the slice. What you do, this is a stimulating electrode. You stimulate, and then you record a cell in here, this cell. Okay, forget about this thing. This is so you get the synaptic activation. Okay, and here's the sizes of those, and then you just don't do anything. But during this time, you make the cell to fire an action potential by just passing a little bit of current during the synaptic activation, after the synaptic activation started, okay, for a while. And then you stop doing the action potentials, and the whole thing gets bigger, okay? So when the action potential occurs shortly after the synaptic activation, you get a potentiation. And the other way around, you make the cell, in this case, to fire an action potential right before the synaptic activation, but before. And in that case, it gets depressed. So it's basically the order, and the only thing that's different between here and here is the same number of pulses, the same number of action potentials. The only difference is the order, who fires first and who fires later. And that's what is called spike timing dependent plasticity. Okay? And actually what you can do now in a more parametric study is to vary the time between the action potentials and the synaptic activation. This would be negative delay, and this would be positive delay. And you can see that when the thing is very short, you can get a lot of potentiation, and as you make it the delay bigger and bigger, you get less and less, and eventually you get nothing, and the other way around too. Okay? So the only thing and this is the cool thing about, and that's what it's called, because the timing codes for the polarity and the magnitude of plasticity. So with this, actually, in principle, you don't need to monitor, how well the NMDA receptor is activated or what is the potential of the cell. The only thing that you have to know is when the presynaptic cells fire and when the postsynaptic cells fire, and in principle, you can compute the potentiation of the depression. Okay? And actually, then you can go to do in vivo recordings in an animal and see, predict, what is going to be a potentiation and a depression without going intracellularly. Okay, so let me see how I can do. Okay, so I have 30 minutes. All right, that's cool. So what is good about this? So the timing codes for polarity and the magnitude, and it's strictly based on correlations, not on the level of activity. Okay, and actually it's a rule that actually encodes causality in a way, because pre then post, it means pre is causing post, that's LTP, and post then pre means that the pre is not causing post, and that's LTD. All right? And so people who do modeling of neural networks love spike time independent plasticity. I mean, this is meant to do modeling of neural network. Okay, is the substance that, okay, and so a lot of people are based their careers on this, and it's very cool. Okay. And synaptic changes can be computed from spike trains. So if the spike trains of different cells that are connected between them are available to you, in principle you can compute what is going to be a potentiation of depression and fulfill the letter of the Hebbian and anti-Hebbian. So people think now that this is actually the way that plasticity really happen in the real brain. The correlations between pre and post synaptic activity, which is translating in strengthening and weakening. Okay, and these things about the low frequencies and the polarization I told you is a much more convenient way to do it in the lab, but it's just a methodological convenience. Okay, that's the. Okay, so how it happened? Well, we know that the pre then post is easy because the AP boosts the activation of the NMDA receptor by reducing the magnesium blocks. That's what I explained you. And here I put several hypotheses how the LTD works. You can read it at your leisure at home. Basically, nobody knows. Okay, so the three alternatives that are not fully proof, and people can go one way or the other. So it's basically we don't know. Okay, so for you for you understanding, the only thing that you should know is that the order is important. You can get LTP and LTD. We can understand how LTP work, and there's a lot of evidence for that. We don't understand fully how LTD work in this context. Okay, so that's all about that. And now we're going to talk about metaplasticity. So we need to regulate synaptic plasticity. Okay, and the reason is because LTP and LTD are intrinsically positive feedback mechanism. Okay, so and people notice that very early when the people start to do neural networks, you randomize the synaptic weight. Some of them are stronger, depending on the condition. Okay, if you start with a network which is slightly inclined to potentiate, well, in the second round you're going to get potentiation of many more synapses, and you're going to get more activity, and the third round you're going to get more activity, and basically everything starts to go up and up and up, and it's going to reach ceiling, and then everybody's going to be potentiated after a few rounds. Okay. On the other hand, if the network was slightly inclined, just randomized to do LTD, well, in the next round, in the next iteration, since you have produced some LTD, you're going to get less activity, and it's going to be more LTD and less activity, and eventually everything will tend to flatten down. So they were very unstable. Okay. So if you let it run by themselves, they're very unstable. So people have to put some feedback mechanism there to stabilize. At the beginning, they're putting just exponential decay. So if everything is going too up, let's put some exponential decay in there, and then we set it backwards. It's going down, let's exponential growth. But that's kind of cheating. So there are two mechanisms actually that provide a good feedback. Okay. So what we need is a negative feedback, such as somehow if you have a lot of neural activity, it will kind of influence negatively in the synaptic response, and if you have and if you have a very little neural activity, you have to kind of pump it up. Okay, and there are two mechanisms that have been proposed, and for which there is evidence. And also, I'm going to show you quickly this experiment that actually you could not explain with LTP and LTD only. And it's about the thing that I was telling you when you close one eye, in this case a little cat. So normally you have the right eye, the left eye, and you stimulate eye, you get responses. Okay, but if you close one eye for 2 days, okay, and then you open again, yes, I don't do that. Okay, don't. And it turned out that the closed eye now is not responsive to stimulation. Okay, so that you can explain that normally with LTD. Okay, so the closed eye have a very little activity, and there was a lack of correlation between this input and the output of the cell, and therefore you have a spike time independent plasticity. It worked cool with the classical LTP and LTD things. Okay, but you can make a variation of the experiment, which is called reverse suture. And the experiment, you started like the same, you close the eye of the poor guy here, okay? But after 2 days, you do something even more perverse. You open that eye and close the other one, okay? The prediction in here, it would be this eye now, it was totally disconnected because the 2 days in the deprivation should have disconnected that eye, okay? And this eye is basically closed. So the guy should become complete. If you are thinking about LTD, this guy should become blind, basically. You disconnect one eye first, and then you should disconnect the other eye, and then that should be very bad, and the guy should get blind. But actually what happened is that somehow it recovered vision, eventually it will recover vision in that eye, and it will lose vision in the other eye. Okay? So with LTP and LTD only, the only prediction it will be the guy will go completely blind. You're very bad. Okay? But actually, it doesn't happen like that. So, and there are two models that can explain that. One is the sliding threshold, and the other one is synaptic scaling. And I'm going to tell you about that. So the sliding threshold, it was developed by these guys, and this is kind of like, it's the same thing, it's the same motif as the Hebbian synapses and the silent synapses of Francis Edwards. These guys developed the model probably good 15 or 20 years before there was experimental evidence for that. And the guy was thinking, okay, well, let's see that this is the function of plasticity. You know, you can put here postsynaptic activity or calcium or NMDA receptor. You know, you have a little bit of activity LTD and a lot of activity LTP. We know that. This is what we have seen it. And then at some point you have a crossover point in here, which everything is equal. Well, what if that crossover point is not fixed? Okay. So what if that crossover point change with the history of activity? Okay. And that can provide the feedback we need. So suppose now that when the cells are there's not much activity, the crossover point goes to the left, to here. So in that case, this point that before didn't produce anything, now it's going to produce more LTP. Okay? And this point in here that it was producing a lot of LTD, now actually is not producing anything, or LTP. So if the system is very low active, you want to promote potentiation basically to bring it back. And one way to do it easy is to move the whole curve to the left. Okay? Because now the same activity that before it was ineffective, now it's going to potentiate and hopefully bring all of the activity back to where it should be. You follow that? And conversely, you know, if the system is super active, like almost epilepsy, well, what you really wanted to do is to cool down and promote LTD. Okay? So you shift the threshold for LTP very much to the right. So it's very difficult to do LTP now. And activity that before produced LTP now is actually producing LTD. Okay? So it's going to shift back to, so that giving a lot of stability. Okay? And then make the whole model and everything that, it works very nice. Okay? And it will explain the thing with the little cat. Okay? Because, well, the idea it will be that the poor kitten in here, you close one eye, and basically the activity of the cortex, in a simple world, it will be reduced by half. And therefore, now the crossover point is going to go to here. And now, the half of the activity now that before it was producing actually LTD, now it's going to produce LTP, so it can come back. Okay, if you activate that. So that's the whole idea. So is that it will provide, you know, like an automatic adjusting mechanism. And actually, it was thinking about this kind of experiment that these guys developed the model. Okay, so that's one model, and this was the neat thing. It was like it was theory before experiment. This other one, it was actually the other way around. It was experiment and then theory. So it's synaptic scaling, and what it is. So it's an experiment, very simple experiment. This was done with the culture cells. So you play this a dish with a lot of cells in there. I think these little dots are cells, and they make a network actually. And this is a network that makes a spontaneous activity. So you can go to a cell and you record, you know, synaptic responses and action potentials and all of this. And then they treat those dishes in two ways. In one, they put TTX to block action potentials. Okay? And that's what they get, nothing, for like 2 or 3 days. In the other one, they add bicurculin. Bicurculin is a blocker of inhibition. So here you have excitatory cells and inhibitory cells, and everybody's acting together, so it's kind of like a balance. But if you block the inhibitory cells, everybody is going to go now to epilepsy. Okay, you have a lot of activity, and you leave it for like 2 or 3 days again. And then you remove the TTX, and then you remove the bicurcurin and measure again. In this case, these guys have been so silent that when you remove the TTX, actually they become epileptic, hyperactive. And this guy that had been totally disinhibited, you remove the blocker and became inhibited. Okay, so something was happening in the network that was counterbalancing the insult, so to speak. So if the manipulation was trying to make the network very silent, well, the network was doing something to become very active. And if you make the network very active, the network was trying to become very inactive to compensate for that. And what was changing, it was actually the size of the synapses. Okay, and it's called synaptic scaling because it turned out that here I have a representation of a pyramidal cells, and this will be synapses. Okay, and if you have low firing rate, there is somehow that's the model say somebody sensed that there's so few action potentials, and you wanted to get more action potentials, so the nucleus probably send a signal to all of the synapses of the cell to increase by a factor. That's the scaling. Okay, make all of the synapses twice as big or something like that. On the other hand, if you have a high firing rate, well, you're going to do the opposite. You're going to reduce the strength of the synapses, so now you're not going to produce so many action potentials. Okay, and you also reduce all of them by the same factor, and that's very important, and that's key, and that's why now is one of the most popular avenues of research on plasticity, how this happened. And the key of this is that if memories are encoded as changing in synaptic weight, what you wanted to do is to reduce all of the synaptic input, but you don't want to erase memories. So you still want to keep the memories in there. And if the memories are coded as relative synaptic weight, if you reduce all of the synapses by the same factor, you're going to keep the same relative weight. Okay? So if this synapse had to be much bigger than this one, and that is crucial for remembering that you have to go to the left to eat, well, and you increase all of the synapses, but this one is still in the same proportion against that one, you still remember. Okay? Because the idea is that it's the relative weight the important, not the absolute weight of the synapse. And so you keep the relative weight of the different synapses. You just change the total strength. Okay, and that's a beautiful mechanism to control the firing of the network without erasing memories, and that was the key thing. Okay. Oh, let me see. We're almost done. Yeah. Huh. I guess I went. Yeah. All right. So, and how that was proved. Okay. So that's the model, and here is the experimental evidence. So what they did is here you have two synapses. Okay. And if you have reduced activity, both of those synapses should go bigger. This is the response. Okay. But you have to maintain the same relative size, and the other way around, if you have increased activity, you have to reduce both, but with the same relative weight. How you can prove that? The trick in here, it was to record the things called spontaneous minis. So what you do, if you put TTX, so there's no any evoke action potential, so you have only spontaneous responses that caused by the spontaneous release of a vesicle. Okay, that happened. I mean, it's very unlikely that happened, but remember that the cell have 10,000 synapses. So even if they have a probability of 1 in 10,000 per second, if you have 10,000 synapses, on average you're going to have 1 per second, and that's more or less what you have. So, but if you wait for a long time, you collect like 1,000 of those. Well, you can get kind of like a good sampling of how the synaptic weights are. And you can also, some of them are going to be bigger, some of them are going to be smaller, so you can contract in here. What they did in here are a distribution of sizes. Okay? Some of them are bigger, some of them are smaller. And the idea is that you have this distribution of sizes, and then and you do a manipulation, and then, for instance, the distribution of sizes go to the left or go to the right. You go to the left, it means they're getting smaller. You go to the right, it means they're getting bigger. But if you have synaptic scaling, this distribution you should multiply by a factor and should obtain the initial distribution. Okay? See, the only thing that you did is all of those synapses, you make it bigger by a factor of 2. Well, you divide this distribution each point by 2, and you get the original distribution. Okay, as simple, and that's what happened. Okay, so these are all of the distribution once you scale them. Okay, so that was so you work on the distribution. You collect like 1,000 or 2,000 events, and in one case, and in the same number of events, the distribution of this is here, the distribution of that is there, but you multiply that by a factor, single factor, you get back to the. So that was the demonstration. Basically, that is all of the synapses are scaling by a single factor. So I went so fast because you didn't ask me questions. Okay. I was expecting some questions. So I'm going to compare here sliding threshold with the synaptic scaling. Okay. So both of them are global in the sense that both of those mechanisms affect every single synapse in the cell. And that's completely different, opposite to LTP and LTD. LTD is completely local. It activates only a subset of synapses that are being activated, okay? And here's everybody, whether you were active or not, okay? And there are experimental evidence for that, and that's kind of cool. So actually, I was working on this. That was my claim to fame and my ticket to Hopkins, actually. So how you can demonstrate that? Well, we have demonstrated in the visual cortex, because there's an easy way to manipulate activity in the visual cortex, which is basically put the guys in the dark. You don't see anything, and therefore you reduce your activity. Okay. So now, if you reduce your activity in the visual cortex, what would be the expectation? It would be that you're going to slide the threshold. Okay. So the threshold should be now, since there's very little activity, it should be much easier to produce LTP, to potentiate it, and more difficult to produce LTD. Okay. And when you bring it back into light, the whole thing chooses. And actually, that happened. Okay. Now, in the same token, dark rearing also, if synaptic scaling work, you should put the animals into the dark. There's going to be little activity, and then all of the synapses should increase to compensate for that, to try to recover the same level of activity. And if you do the experiment, actually, that happened too. Okay, so there's evidence for both of them. And one of the interesting things is that the sliding threshold doesn't affect the stored memories, because it just changed how new memories are going to be acquired, but not all of them. And this one, synaptic scaling, doesn't affect stored memories, because actually it's maintaining the relative size of the synapses. Okay, it's just the absolute value that is changed. So they're very similar, and actually the big question is whether perhaps they're just two sides of the same coin. Okay, and that's the big interesting question that people ask now and then, whether actually they're not just the same thing in a way. And why is that? Well, because in the sliding threshold, you put the animal into the dark, and there is not much activity, okay? But you are going initially, but you're gonna facilitate LTP now, okay? So now you start to get LTP, okay? And eventually, the synapses are gonna get bigger, so you can get synaptic, which is what you find in synaptic scaling. So you can imagine some scenarios in which that can happen. So you can get an increase on the synapse weight just out of by putting the animals in the dark, and assuming some spontaneous activity. Okay, all right. The other way around, it can also work. You put the animal into the dark, and now the synapse start to jacking up. Okay. Now, if you have a bigger synapse, now it's going to be easy, more ample receptors. It's going to be easy to activate the NMDA receptors, because now you're going to get more depolarization for the same amount of input. Okay, so that would explain why it's easy to get LTP. So both of them can explain the other one. And at some point I asked Cooper. Cooper is the guy who developed this model, and actually he got the Nobel Prize for the famous Cooper pairs electrons. Okay, so he was working in superconductivity, very bright guy. He get the Nobel Prize for that, and he said, well, I'm going to start neuroscience now because there's no more matching superconductivity to be done. And he developed this model, and I asked him, about these two things. And he said, actually, he doesn't care, okay? Because it's basically kind of like an axiom. Sometimes you can have an axiom that you can go from one axiom to the other one and vice versa, and it's basically, for people like him, those are probably just the two sides of the same coin, okay? But phenomenologically, you study them in very different ways, okay? So that's it's still convenient to separate. And actually, we don't know whether they're actually two different things or not, okay? So I think I'm going to quickly recapitulate. So the first part of the talk, it was introducing you to the notion of spike time independent plasticity, in which the idea is that the pre and the postsynaptic spike, the timing between them and the temporal relationship is the primary determinant of the polarity and the magnitude of plasticity, and the only thing that in principle you need to know. And that's probably a better model of how it really happened. And the second one is that you need these mechanisms of homeostasis to maintain, to check, for LTP and LTD to make them, more stable and not crazy. Okay? And all right. Any question? Not a single one? Ah, okay, good.
What do you mean by having bigger synapse? Is it like the physical size of the synapse?
It's actually the number of AMPA receptors and eventually the physical size. So those responses become bigger, okay? The deflection that you measure it became bigger, and that's because you have more AMPA receptors in the synapse. And eventually, when you get more AMPA, you have to grow on the size because you have to make room for them. So it's both. Electrically, you can see the electrical deflection, the current, okay? And also, when you do imaging, you can see them bigger, okay? So it's basically more AMPA receptors. And actually, I should have mentioned, and I forgot, in the sliding threshold, what they change is actually the NMDA receptors. When you go into the dark, you put a new kind of NMDA receptor. It's called NR2B that have a longer duration. So normally the NMDA receptors remain active for a while, for like 50 milliseconds. And this NMDA receptor now is going to remain for like 300 milliseconds. So you can get more calcium, basically, for the same amount of NMDA receptor. And that's a way to slide the threshold. Okay. All right? Okay.